Hello, my name is Anika, and today I'll be presenting on Leighs syndrome and the impact it has on the nervous system. So, Leighs syndrome is a severe neurometabolic disorder that typically becomes apparent in the first year of life, and it's a genetic disease that can be caused by mutations in nuclear DNA inherited from both parents or mitochondrial DNA inherited exclusively from the mother. But regardless of the source, the result is a progressive decline in movement and cognitive function, and it mostly affects the basal ganglia as it's a very energy-demanding part of the brain. Neurons are the body's most energy demanding cells. Usually, in an unaffected person, a massive portion of cellular ATP is used to power the sodium potassium pumps, which constantly move ions against their concentration gradients, which are essential for maintaining the membrane potential. And this allows neurons to fire action potentials and communicate with one another. So the basal ganglia and brainstem are very ATP demanding, so they suffer, and this leads to cell death. So mitochondria are organelles that produce ATP as usable energy by converting glucose into ATP. So while the nucleus codes for most protein, 13 essential proteins are coded by mitochondrial DNA. And a key concept for Leighs syndrome is heteroplasmy, so the mix of healthy and mutated DNA within a single cell. Because a cell has so many mitochondria, the severity of the disease really depends on the ratio of broken to working DNA. And because the symptoms only appear once a certain percentage of mitochondria are dysfunctional, the severity of Leighs syndrome can vary between individuals, even with the same mutations. And there are like 75 to 100 gene mutations linked to Leighs syndrome. In a healthy individual, cellular respiration converts glucose into ATP through these four stages. And the cycle produces electron carriers, NADH and FADH2, which are needed for the final stage, oxidative phosphorylation to convert ATP into usable energy. And Leighs syndrome primarily disrupts this last stage, oxidative phosphorylation. This is a stage where a mass majority of our ATP is generated through the metabolic process. In Leighs syndrome patients, the drop in ATP means neurons can no longer maintain those critical ion gradients, which eventually lead to cell death. And the 13 mtDNA proteins really affect complexes 1, 2, 3, 4, and ATP synthase, which is in our electron transport chain. So specifically in the electron transport chain, the ETC, the most common mutations affect complex 1, the largest complex which accepts electrons from NADH, or complex 4, where electrons are passed to oxygen. So these genetic mutations alter the primary structure of these proteins, and this change in the sequence disrupts the folding and tertiary structures, rendering these enzymes, the enzyme complex, non-functional. And when these complexes fail, the proton gradient here is not established, which means that the ATP synthase, the molecular motor, is unable to spin and be fully converted into ATP. So because it's a metabolic process, Leighs syndrome creates lactic acid, because the genetic mutations impair this mitochondrial energy production, which forces the cells to rely on inefficient anaerobic metabolism. When the mitochondria cannot properly use oxygen to break down fuel, pyruvate builds up and it converts into lactate, and this causes lactic acidosis. And this is a common symptom for Leighs syndrome patients. So in summary, Leighs syndrome is a genetic failure of the mitochondria. Mutations lead to misfolded proteins in the ETC, preventing the creation of a proton gradient. Without ATP, the high-stakes active transport in a neuron fails, and this leads to cell death in most metabolically active parts of our brain. These are the sources I used, and thank you for listening.